at an alternative adjuvant to combine with that to get an even greater T cell boost. So we've taken perhaps a slightly schizophrenic or shotgun approach because I'm not sure we know today which one is the winner or whether there is a winner. And I think to Dr. Fauci's slide, you have multiple different approaches that are in play, hopefully all of which will succeed, because I think the more options we have, the better it'll be for us in the future.
Thank you. You know, we talk a lot about the benefits of having multiple shots on goal, to use one of our sports analogies. And so it's interesting to hear the different ways in which you guys are approaching that, Sandeep. So thank you for that. Let's turn to another question. This one will be for Angela and for David. The historic public-private partnerships we've talked about this morning a little bit already between the government and industry led to the development of first-generation vaccines on a timeline not previously seen. As we think about the development of the next generation vaccine and overcoming these challenges that we're talking about with this pandemic, the ever-changing variant compilations, what role do you see for the USG as we move forward? Angela, why don't we start with you and then we'll turn to David.
Well, thank you for the question, and also thank you for having all of us here today. Pfizer has just been honored to be in this journey together with the U.S. government and with the world. And we are, just deeply committed to finding solutions and continuing to be part of the solutions for the world. We've learned a lot from the time that we've had over the last several years. And I want to say the fundamental to our ability to bring this vaccine forward was an incredible partnership with the U.S. government. But I would say that there are some areas of learnings that I think we can build off of. As we've talked about today, to be able to solve a problem this big, this massive, it really takes just a tremendous amount of stakeholders and a tremendous amount of commitment, investment. We talked about investments at risk today, incredible innovation. But I also think that there are other enablers that really, I think, came to the forefront. And first, I will point to what we began. Dr. Fauci began his talk today about the incredible science that exists and how that needs to lead the way. And in order for us to continue to do that, we have to ensure a thriving innovation ecosystem. That means that we need to have protection, intellectual property rights, intellectual property respect, so that scientists and industry can continue to do their good work. So that's fundamental, fundamental sort of like cost of entry into this. But I think we learned a lot as well from the time that we've had and how we were able to bring a vaccine into the market at such unprecedented speed. And first, I would say that there is a learning and a continued work that we can do together to ensure that there is a fit-for-purpose regulatory approval process, right? So we can have a great drug, a great therapy, but if there's no way to get it onto the market and improved quickly, that does not help patients and does not help the public. So I think the flexibility and the fit-for-purpose way of looking at what is the issue at hand and what is the kind of approval that we need, I think is one key enabler. Secondly, the clinical trial process and the actual conducting of the trials and the recruitment of the trials was another area where speed really helped us. And I want to really talk about the use of digital technologies and virtual tools that enabled us to recruit at incredible speeds, but also to recruit in diverse communities, because we wanted to make sure that at least 30% of our clinical trials for community was represented by minorities. And in this instance, we were able to use telehealth consults. We were able to deliver to our patients' homes the product, the clinical trial product. We were able to conduct virtual visits and follow-ups. And all of this meant that we were able to bring the trial to the patient rather than have the patient come to a site that may be too far. And that's how we were able to increase the number of patients that we had hoped to have rather quickly. In fact, in just over 100 days, we recruited 47,000 patients, which is sort of unprecedented in the short time that we had. But I think what really helped us there was the application of technology and the ability to bring the trial to the patient's home. So I think that would be another area that we can continue to partner on and to build off of. Importantly as well, I want to say that the kinds of trial design was another enabler of speed. And here I want to point to adaptive clinical trial design. Typically you would go through the phase 1, phase 2, phase 3, but in this moment, we really didn't have the time to do that. So there's the incredible willingness of the FDA and other regulatory agencies around the world to partner with us and to really have robust conversations around what are the pre-specified endpoints and outcomes that we expect to see by when, and what are the safety measures and bar that we all want to aspire to be able to have that kind of discussion way up front and be able to then adapt our clinical trial designs according to that is what enabled us to get to the end quickly. So I think it would be great to be able to see the application of this kind of trial design beyond what we did just for the first COVID vaccine, but to other possible vaccines and to other possible therapies as well.
Angela, thank you for that. It is interesting. One of the lessons we don't want to lose in this response are those adaptations we made together, where we were able to adjust quickly to be able to provide this very safe vaccine through clinical trial adaptation and the other pieces that you mentioned. So we want to make sure that we don't lose sight of that as we head into the next generation, very important work. Now, David Kessler, you've been part of the government's response for the last several years. So you've seen this firsthand. What would you say is the role of the U.S. government moving forward with the next generation work?
To date, because of the effort of all of you, the collective we, have delivered 784 million doses of vaccine safely. As of today, there are 601 million doses have gone in arms. That is historic. Go on, all of you, take a bow. I think that's the context. I think we recognize, we've heard this morning, there is a consensus that in light of all of that, that we need vaccines with longer duration and greater breadth of protection. I think we have that consensus. No one is, resting where we are. Our colleagues at the VRC and in industry, certainly just intramurally, as Dr. Fauci has said, extramurally, our colleagues are working, now on intranasal delivery to better protect against transmission, nanoparticle vaccines that include multiple antigenic determinants. That is underway. There is, from all of that gone on, there is an incredible team. You live at Dawn, your colleagues at ASPR, at BARDA, at FDA, at NIH, at CDC, at industry, at DOD, that are poised to overcome the challenges, develop the robust pipeline to support advanced development. I think we have to get these vaccines out there in the next generation. So we are exploring, the processes of going commercial, right, while still ensuring access for the and underinsured. It's a very important thing that we have to continue to achieve. I think bottom line, there is an exceptionally strong case, for investment, right? I think both, the Congress, through their wisdom in the past, the capital markets, industry, I think that continues and really is essential.
David, thank you. And thank you for those reflections on the work that we've done and the work that needs to come and how energized the U.S. government is to see this work through. So, Paul, this question is for you. In the US and globally, we've expanded manufacturing capacity to support global scale manufacturing of COVID-19 vaccines, including critical components necessary for manufacturing. From an industry perspective, how do we maintain this capacity for current vaccines, next generation vaccines, and for future pandemics?
That's a great question, and thank you, Secretary Connor. It's a privilege to be here to represent Moderna today. Look, I think distributed manufacturing, which is really decentralized manufacturing, is a key part of the future. For Moderna, we have announced recently collaborations with governments in Australia, the United Kingdom, Canada, Kenya as well, where we will actually be able to build plants, manufacturing facilities, R&D facilities in those countries, make product for that country, in that country, by that country. It's all part of an important ecosystem of health security, which I think many of the vaccines that we're fortunate enough to have today can bring to population. So that's very important. We certainly partner with manufacturing organizations, contract manufacturing organizations around the world. As vaccine technology continues to evolve, production of vaccines becomes more and more technically difficult, but the size of the facilities that we need to manufacture actually becomes smaller. mRNA is highly reproducible, can be made with high fidelity in very small manufacturing kits, but we can collaborate with those manufacturers. And also here in the United States, we've expanded manufacturing in our Norwood facility. So those are really the technical approaches that we're trying to bring for that decentralized manufacturing around the world. And I think the theme of all of the discussions is, we have to continue these discussions, continue these collaborations to instill confidence for people who at this critical juncture, critical part of the pandemic, have to have confidence, as Dr. Kessler said, 600 million doses in this country given alone. We know everything really there is to know about the safety profile of these amazing medicines. So getting that confidence and then the ability to get these shots made in countries and into people's arms, I think is how we're going to continue to get out of the pandemic through this endemic phase and then into the next generation of manufacture.
And, it's just an important point to talk about all you all have done globally to bring this forward and how important this capacity is for all parts of the world. And so, the investments we're making now, we will have to leverage moving forward. And unfortunately, it looks like we're having multiple opportunities to do that. So thank you for that, Paul. Appreciate it. We have some questions from the audience. So I want to make sure that we get these in while we have the opportunity. Now, this question's a little provocative, but I'm going to go ahead and ask it, and then we'll kick it over to our panelists. And we don't have the benefit of FDA on this panel. We have the benefit of a former FDA commissioner, Dr. Kessler, but you don't need to speak on behalf of FDA, of course. The question is, how long will it take for these vaccines to get to licensure? Now, without having an FDA person on the panel to respond to exactly how FDA might approach this, what I'd encourage our panelists to talk about is the way in which you would accelerate getting your data forward, doing your clinical trials quickly, so FDA at the proper time would be able to evaluate it. So Paul, why don't we start with you?
Yep, thanks, Dawn. I can say just a few comments. So I think transparency of data is fundamental to everything. Getting it out into publications, preprint servers today, describing it publicly is key. That gives people confidence, continues to give them confidence. I would say that we were able to go from the recent VRBPAC meeting to actually manufacturing a vaccine today that contains BA.4/5, and that was probably under a month ago. So I think we can manufacture very quickly. We can get into animal studies quite quickly. I would say the debate now that will go on in a august community like this and socially is, what degree then of animal data do we need versus human trial clinical data do we need? That will certainly determine how quickly we can get to application and licensure. But throughout the whole pandemic, I think what we've been able to do is really partner with regulators around the world, certainly the FDA here in the US. And we've learned so much about rapidly, but with great quality and certainly never sacrificing safety, how we really can speed these applications down to weeks.
Maybe, what is your perspective here?
So I think the theme of this summit is really about partnership, and it's about engaging early together with the regulatory authorities to understand what's needed, right? Especially in this changing dynamic. The situation is constantly evolving, and to be flexible and to be reactive and to be adaptive to that, I think is incredibly important, especially for a small company like us to know what does the agency want to see? What do they need to see? And to know that upfront as best as we can. You know, LMICs are a core part of our strategy. And so we're working with multiple globally recognized regulators, both high income and low and middle income, on accelerated approval pathways. And, you know, I think engagement is the critical path, and being in front and having an open discourse. That's at least our wish. And I think the FDA has done a fantastic job, obviously, since the beginning of 2020 in doing that. So I would hope that it would continue.
Great. Thank you, May. Angela, how would Pfizer approach this?
Well, I think the theme, you know, we've talked about here a lot already, and I just want to, you know, I agree and concur that engagement, constant dialogue, there's no substitute for that, right? An understanding of what are the agency's goals, what are the requirements, and then how can we meet them in a flexible and creative way, especially in a time of the pandemic. The only way you can do that is to do that, you know, constantly with each other. So I can't underscore the importance of a continued and excellent and collaborative, you know, relationship, which I do believe we've had, and is actually you know, we reap the success of that relationship that we've had, and the scientific exchange has been, you know, so vital. The other thing is what I mentioned, which is, as a company, what are the ways and how can we reimagine how work can be done to cut out white space and to be able to bring the same outcomes, but in faster, right? So the application of technology, digital, you know, new protocols, the examples that I've used previously are the ways that we have and we will continue to innovate around to ensure that we can get out there faster and better. But maybe one thing we haven't talked about today is the importance of real-world evidence. Today, you know, we've just talked about how many people in America have had the vaccine, and actually around the world, probably 2.5 billion people have received the Pfizer vaccine. That's an incredible wealth of real-world evidence that we're sitting on. And so I think that we have a great opportunity having that strong depth and breadth of data to use that to also help us to understand and how can we design new therapies, but also how can we apply it to the public in a you know, to facilitate, to expedite, and to be able to gain access quickly so that, you know, the public can really benefit from this.
Thank you. That's a really good point. Sandeep, how would you all approach this?
Yeah, I mean, I think obviously I agree with my colleagues, but I think one of the most important and really pressing issues is identifying what would be appropriate correlates of protection that regulatory agencies would be willing to accept for approval. It's very clear that that actually is something that is at the forefront in other jurisdictions. So for example, in India, there's a vaccine that was approved based literally on scientific data showing that it generates a sufficient immunologic response. And, you know, when we're talking about shortening these timeframes, I think the reality is in a world where, you know, you guys can tell me how many 100 million people or billion people have been vaccinated, right? 2.5 billion at least. It's going to be very difficult to do a traditional endpoint type study. But it's very easy and very rapid to do a correlates of protection type study. And for us, I think we look at it not just in terms of humoral response, but also cellular mediated response. What is a sufficient and clear goal? You know, what's the goalpost that we need to hit in industry? If that was known, that would make it very easy to design those trials. And I think going forward, that's going to be critical to get us to where we need to be. When we think about, and I'll just pick up one of the points that Paul made, when we think about, for example, self-amplifying RNA, from a manufacturing perspective, we don't need massive scale. We can do this, but what we don't have is unlimited number of patients. And so having some alternative way to be able to identify success and identify success rapidly, I think that's a very critical need at this juncture.
Thank you for that. David, any additional thoughts or comments?
I am absolutely 100% confident to the leadership of FDA. I think we have seen they have risen to the occasion. They are creative, they are nimble when it comes to these vaccines. I have a great deal of confidence. I do think there are hard questions ahead of us, right? With the vaccines, with the effectiveness, the very high effectiveness against serious disease. What is, quote, better going forward? Is a hard question. I think we're getting closer on not necessarily a perfect correlate, but with correlates that will help us. I am concerned. I think one of the big impediments over the last several years is I think we have been, I am enormously grateful for our international colleagues, but for the Israeli data, but for the UK data, right? That has been essential. I think we are too reliant, on some of the international, they're great. But your point, Angela, on real world evidence data, that very strong epidemiological data, I think is absolutely critical. And we have a long way to go to develop those systems.
Very good points. Another question from the audience. One of the historical challenges for vaccines is the mismatch between the money needed to develop them and the ability of those who need them to pay. How do you foresee a path forward for public-private partnership that threads this needle? So we've talked already about the public-private partnerships and the need for that, but how do we manage this piece? Anyone want to try to take a first stab? Angela.
We've had tremendous learnings from the distribution of the Pfizer vaccine into the developing world. And if I think about the lessons learned, I think you can look at them sort of in two chunks. The first is, how do you get the vaccine distributed there? And then secondly, you have to look at how do you actually get the vaccine used? And I think on the first part, the ability to supply, we were able to meet that challenge, and we were able to meet that challenge really well. In fact, of all the vaccines that were distributed in the world in 2021, 1 billion of that went to the developing world. So I think in terms of equitable distribution and the ability for people to access it, they were able to do that. I think what we learned, unfortunately, is that the ability for these countries to utilize them was a different thing. And there are large issues related to vaccine confidence, misinformation, ability just to have, enough healthcare professionals administer the vaccine. And so if you look at sort of the last mile challenges and the complexity of what that takes for someone to actually get the vaccine, I think that we did not spend enough time there. And so one of the things that is going to be really important, well, has become important, but will continue to be important in order for everyone in the world to receive the vaccine, is healthcare system strengthening. It's everywhere. I would say that's in parts of our country, but also very, very, visibly in the developing world. And it's one of the reasons why, as a company, we have advanced on our thinking, and we are in an agreement and in partnership with several African countries, actually as part of a different initiative that we have launched called An Accord for the Healthier World, which goes beyond vaccines. It's about making all of Pfizer's innovative products available at a non-for-profit price to developing world and to the low-income countries. Part of that is not just about distributing and dropping the medicines off. A big part of that is about understanding what is needed for capacity building, healthcare system strengthening, and bringing together, talk about partners, more partners in these countries, so that we can really understand what it takes to be able to administer and receive the vaccine, not just supply it. So I think the supply part was something we understood really well. I think the remainder last mile part is something that we need, I think, the entire world to focus more on.
Absolutely. Paul, did you want to jump in?
Just very quickly, Don, I know there's going to be a panel later on equity as well. Clearly, we partner with COVAX, CEPI, Gavi, other major institutions around the world. But I think just bring it closer to home, Medicare will clearly take care of people over 65. There's the Vaccines for Children initiative for those younger kids. I think the people between 18 and 65, those underinsured, uninsured, is very important. And I would just call out the initiative of the president, the Vaccines for Adults initiative, which I think is extremely insightful, providing access there for vaccines that would be ACIP endorsed, I think is a tremendous step forward in access.
Absolutely. Thank you, Paul. May?
Yeah, so I think when problems are global, we have to proactively address them as global and not have the rest of the world as an afterthought, right? When you think of it as a global launch, I think you prepare and think about all the things that need to happen. Fortunately, vaccines are one of the most prolific medicines in our history, right? Over 80% of the people, even pre-COVID, were vaccinated. And because of that prolificness and that accessibility, they've been one of the most impactful medicines, probably doubling our life expectancy over the last 100+ years. I think technology really is an enabler there. So yes, we can find a vaccine that works, but can it be scalable? Can it be cost effective enough that it can get to and can be affordable? And can it be distributable, right? So can you make it so that it doesn't require cold chain storage? Can you make it so that it can survive anywhere a Coke can go, right? To every part. And I think the technology is really has historically been our equalizer and our enabler. And that's where we should look at to solve these problems. Technology has always equaled equity. Look at the printing press, right? Prior to the printing press, books were the most expensive commodity on earth. They were more expensive than gold by weight. It's because they had to be handmade and monks had to literally write them. Then the printing press came around and we democratized the book, right? I think that's where technology comes in. So how do we democratize vaccines? And I think we're on our way, but ultimately technology has always helped us make that exponential leap to create equity across the globe. So that's where I would focus on new.
Thank you, May. Sandeep, did you want to get into?
Yeah, I think so. I mean, first of all, I agree with what particularly what May just said. I think it's interesting. So we've been fortunate enough to put together about 1 million square feet of manufacturing capacity without taking any public money. And the reason is that we started, again, as I mentioned earlier, as a cancer vaccine company, so we were able to pivot. And really the underlying point is that there is capacity. There's significant capacity. And the question is, what is it used for, right? So that's number one. Number two, the ability to produce vaccines at scale at low cost is not an issue, right? I mean, that's, as May pointed out, the majority of the world is vaccinated against a variety of different diseases. So we can do it. It's just, what is the right vaccine and how fast can we get it out there? Now, as we move into this next generation of vaccines, what's exciting is the ability to possibly lyophilize these and take us out of cold chain. I think, and I don't want to steal from the upcoming panels, but it'll be very interesting to hear about mucosal delivery. I think getting IgA and getting reduced transmissibility would be very exciting. If we could get into that type of environment where a lower dose can be used, right? A much lower dose. Now the scalability becomes very approachable, right? The COGS go down dramatically. If there's a several log reduction in the dose, there's a several log reduction in what we need to do to manufacture it. So the underlying point is that absolutely technology is here. I don't think it's coming. I think it's here. And we just have to apply that technology appropriately. So it'll be very interesting to hear about vaccine equity, but I think we have the solutions. We just have to implement them correctly.
Wonderful. David, did you want to comment on this?
Yeah. Can I just to underscore a point that Dr. Fauci made earlier? You know, the investment didn't begin for COVID in 2020. Investment began for COVID, right? You go back Moderna years ago, BioNTech, Pfizer years ago, the NIH, you know, a decade plus. That's what we built. You know, the current response. That's what we have to recognize, right? And that investment in that basic research through the American people, through their Congress, right? You can't just pull a vaccine out today when you need it. You have to pull it, as my colleagues have taught me well, based on all that science that has gone in the years past. That's what the investment has to be.
So I'm going to do something unusual for our last question. We're down to our last 2 minutes. I'd like each panelist to tell me what you think the benefit of this next generation vaccine is going to be to the American people, to the world, if we can land it. What will it mean to people? People are exhausted. They're tired of COVID. They've been struggling with this for 2.5 years. If we can get this next generation vaccine, what will that mean? 1 or 2 words, 1 or 2 sentences, and we'll just keep moving down the line. Paul, start with you.
Well, look, I think it will give confidence and a way to finally leapfrog the virus, get out of the pandemic phase, truly into endemic phase, and cement a foundation for coming pandemics, which sadly, as Dr. Fauci and others said earlier, will doubtless be a part of the future.
May, what will this next generation vaccine mean to America and to the world?
I think it'll set an example to folks that we can do it and that we can tackle other diseases plaguing humanity, right? That we can tackle scientific problems together, that we can build trust with the public successfully, that we can save lives by working collaboratively, and that we can set up new platform technologies that can then be leveraged for other diseases. And like I said, you know, we're working on COVID vaccine, but that investment is broader than that. It can, you know, we're working on vaccinating for Alzheimer's, migraine, heart attack, and stroke. These things are not just singular in their impact. They build on each other. They're exponential, and I think that showing an example for that will mean a lot for everyone.
Angela, what will this next generation vaccine mean?
I think that the ability to create a next generation vaccine will just demonstrate once again that as a country, as an industry, and as a scientific community, we have the tools, we have the expertise and the ability to rise to the challenge, whatever that challenge is. And so that should create a great deal of confidence and security in who we are as a scientific community. I think importantly for all of us who are just citizens, I think just relief, healthy living, the ability to conduct life in the way that we want.
Sandeep, what will this mean?
Yeah, I mean, I think normalcy, which whatever that means, right? Whatever that means, but also, I think ultimately cross-reactivity and memory from a vaccine will give us the confidence to, you know, if not just this pandemic, but as Dr. Fauci mentioned, we've had 3 essentially in the last 20 years. But let's get rid of this pandemic, but the next one too, and the next one and the next one. And if we have that ability to prevent, that's really what we want out of a vaccine is prevention.
David, what will this mean?
I think the current vaccines have shown that they can prevent. You don't have to die. You don't have to be seriously end up in an ICU. I think that's what the current vaccines have done. But we don't know, you know, how this virus may continue to mutate and come back in a stronger form. And I think a next generation vaccine will safeguard and be able to allow us to avoid the death and disability from COVID in the long run.
Well, thank you. And I want to thank all of the panelists. You can tell how lucky we are to have such strong partnerships with innovative thinkers, people who are facing this complexity with vigor and creativity and, you know, a desire to get us out of this pandemic and to help us get to this next generation vaccine. So I want to thank each of the panelists, want to thank the audience for the questions that we received. It was terrific to hear from you, to know what was on your mind. And I think we're going to break now for lunch. And those of you that are live streaming, we'll be back at 1:15. Thank you, everybody.
Privileged to be the moderator of this second panel. We're going to talk about innovation in vaccine delivery. And so I'm going to be heard from my wife about how it's possible we turn up 3 days of this. So we have a distinguished. We're going to have fun talking about the science of vaccine delivery. I'm going to take off my mask now, and since we are supposedly going to be articulating in a particularly clear and coherent way. And I have with me people who are going to fill you in with some of the most interesting developments that are happening. In Tony Fauci's really remarkable summary this morning, he talked about 2 major ways in which we can see the future of vaccines for COVID evolving. One being the way in which one can come up with ways to immunize against common antigens, or perhaps do the major multiplex mosaic thing. But the other thing he mentioned was mucosal administration, which happens to be not the only way that we might come up with vaccine deliveries that are not the same as what we're all used to with needles into arms. And we are going to hear about that from this group, both in terms of nasal and also microneedles. And we also have distinguished basic science expertise about the immune system and particularly how it relates to this topic. And we're also going to have the chance to have a real world estimate about how this would all be seen as a useful addition to the public's opportunities to take advantage of vaccines. So let me quickly introduce the speakers who are here in alphabetical order, and then I will ask each of them to speak for exactly 4 minutes, or less would be all right. And then we'll engage in a bit of a conversation about issues. And then we're ready also to take your questions. I've already gotten a couple of questions that have been submitted. Maybe there'll be others in the course of the next little bit. So next to me is Dr. Miao He, who is the founder and chief executive officer of CyanVac LLC. Next to him, Dr. Akiko Iwasaki, who is the Sterling Professor of Immunology and Molecular, Cellular, and Developmental Biology at Yale University. Next, Marty Moore, who is founder and chief scientific officer of MISA Vaccines. Next, Mark Prausnitz, chief scientific officer of Micron Biomedical. And finally, Michelle Smith, director of the Office of Health Equity and HIV Elimination, Arkansas Department of Health. I couldn't help but notice in looking at the bios that 3 of our members of the panel have connections with Georgia, and I don't know what to say about that. I just had to comment. University of Georgia, Emory, and Georgia Tech are all represented here in various ways. So something about their ability to do innovative things, or maybe having the CDC next door has something to do with it. Anyway, welcome to all of you. I'm going to go in a particular order because I think it might build this way from sort of the basic science to the public application. So first, I'm going to ask Dr. Iwasaki to give us her 4 minutes of what is most important to get this conversation going. And you may need to get close to the microphones. I noticed in the first panel that if people were a bit far back, it was hard to hear because I was sitting in the back row. So please proceed.
Hi, my name is Akiko Iwasaki. Can you hear me in the back? Okay, wonderful. Thank you. So I'm a professor of immunobiology at the Yale University School of Medicine and an investigator at the Howard Hughes Medical Institute. I've spent most of my scientific career studying mucosal immunity and developing mucosal vaccines against viruses. I'm here today to talk about nasal vaccines to help end this pandemic. And so, as you know, SARS-CoV-2 is a virus that's transmitted in the air. We inhale these viral particles. They land on our nose, our mouth, our throat, and in the lung, and start the replication process. Currently, the conventional vaccines do not induce mucosal immunity, although it induces very robust systemic immune responses that circulate in the bloodstream. Okay. So decades of research have shown that mucosal immunity requires mucosal vaccination. So mucosal vaccines induce local immune responses that are compartmentalized and specialized, such as IgA that's secreted into the mucus layer, and it can capture the virus before it even enters the host cell. Even if that fails, there are tissue-resident memory T cells and B cells that can be established in these mucosal organs to quickly eliminate infected cells before the virus has a chance to spread. And so this was result in reducing not only infection, but transmission as well as reducing the burden of disease within the infected individual, because we're stopping the virus from spreading right at the border. This is akin to putting a guard outside of the house in order to patrol for invaders, compared to putting the guards in the hallway of a building in the hope that they would capture the invader. So how do we elicit these mucosal immune responses? Well, most licensed vaccines that are used in humans are currently live attenuated pathogens, which cannot be used in immunocompromised individuals. However, we came up with a new strategy called Prime and Spike, which leverages the immune responses elicited by conventional vaccines like the mRNA vaccine, and convert that into a mucosal immunity by spraying into the nose recombinant spike protein or mRNA encoding the spike protein that's encapsulated into immunosilent nanoparticles. Using this prime and spike strategy, we are able to inhibit infection, transmission, as well as disease burden in animal models. I'm a co-founder of a company called Xanadu Bio, which is trying to raise enough funds in order to take this to human clinical trial. However, we are not there yet. It's still very early stage. So I'm really here to talk about, the need for the collaboration between the government, the private sector, and the academia in order to bring these innovative strategies forward as quickly as possible. And you will hear other creative strategies from the panelists here today, but it's very important to note that we need to move quickly to start testing these nasal vaccines in humans, and that requires a significant U.S. government input, both resource and help with manufacturing and delivery, as well as acceptance across the society. So I'm really happy to be here on the panel.
Thank you very much. Now let's hear about two of the different approaches to nasal vaccines. I'll begin with Dr. He. Please tell us about what CyanVac is doing.
Hello, my name is Biao. So in case you're intimidated by 3 vowels in my name, it's pronounced meow with a V. So don't forget V with meow with a V. So first, I'd like to thank the organizers for the invitation. I'm really excited to be here to tell you about the progress we have been making to develop a human intranasal vaccine platform. I am Fred Davidson Chair of Veterinary Medicine at the University of Georgia College of Veterinary Medicine. And as well as Dr. Collins mentioned, I am the founder and CEO of CyanVac. And Dr. Fauci mentioned, and Dr. Iwasaki mentioned as well, the infection of many pathogens to human came through respiratory tract, which often means nose. And then we really need to find a way to stop the virus where it enters us. So for past 25 plus years, we have been developing a human intranasal vaccine system uses a nasal spray to deliver an antigen, to immunize people. So I look around, I suspect many of you have dogs at home. And if you have dogs, you most likely have your dogs vaccinated with the kennel cough vaccine. So kennel cough vaccine is given to your dogs through dripping the vaccine into the nose of your dogs. So in the dogs who have been using this vaccine for over 40 some years now. So in past 20 some years, so we have been essentially to re-engineer the component of this kennel cough vaccine. So we repurposed the virus in this kennel cough vaccine called the parainfluenza virus 5, PIV5, to use it as a human intranasal vaccine platform. So using this human intranasal vaccine platform, we have, of course, developed many vaccine for nasal inoculation, for instance, respiratory syncytial virus, and of course, most recently, we pivot to do COVID-19 virus vaccine. So the vaccine is right now in human clinical trial in the US. So we're very excited to be here and to tell you about what we do in this vaccine and this platform system. And last but not least, I really want to thank my collaborators and supporters over the years to make us go this far. One of the places I really want to thank to is NIH. So, it's as they mentioned, and I think Dr. Kessler and Dr. Fauci mentioned, if you look at the slide, I'm very happy he put our company on one of the intranasal vaccine that's supported by NIH. Because over the years, when I started as an assistant professor, I had this idea. Back then, I didn't know our vaccine vector would really work for whatever we're doing now, right? We got funding from NIH. NIAID mostly supported us for over the years. So I'm really, really grateful. So I'm here and I'm excited about what we're doing, and I'm very hopeful and very optimistic that our intranasal vaccine platform system, as well as our intranasal COVID-19 vaccine, will contribute to control of the COVID-19 virus infection. So I will stop here.
Thanks very much. Dr. Marty Moore, another representative of a company that's in the leading role here trying to develop intranasal vaccines at MISA. Tell us about your approach.
Thank you. Yes, my name is Marty Moore. I'm a PhD virologist, co-founder and chief scientific officer of, it's pronounced MESA Vaccines,
Thank you.
which is a clinical vaccine biotech. So, the pandemic really exposed the vulnerability of America's health and way of life to viruses. As we heard today, fortunately, life-saving injectable vaccines were rapidly and successfully deployed, saving over 2 million lives. So why are we here talking about the need for new, different vaccines? Let me paint you a picture of two futures. The first future is the road we're on, where the vaccine is essentially transmitting and spreading unchecked. And this drives the evolution of new variants and outbreaks. The virus is mutating faster than our ability to manage vaccines, and not enough people are getting boosters. These injectable vaccines do a great job at preventing severe disease, but they do little to prevent infection and transmission is the engine of variant evolution. So in this future, right, COVID is a consists of rolling outbreaks of disease disruption, and the alternate future. So what's the alternate future? Well, the alternate future is based on a successful roadmap. The path we took with polio virus, okay? The first polio vaccine, like the Operation Warp Speed of its day, was injectable, was rapidly developed. It worked and it saved lives, but that wasn't enough for polio, and it's not going to be enough for COVID-19 because vaccines that you administer with a needle don't prevent viruses like this from entering your body. To do that, you need mucosal immunity, as Akiko said, okay? So subsequently, the generation of a second generation polio vaccine did this. It provided this frontline immunity, it reduced transmission, and this was the key to controlling polio globally. We can follow in these footsteps. We can develop transmission blocking COVID-19 vaccines as an end-game strategy. How doable is this? What do we really know about mucosal immunity and COVID? Well, we're still learning. We know that SARS-CoV-2 infection itself can prevent reinfection. It actually does so better than injectable vaccines. We know that infection stimulates mucosal immune responses in your nose, and that natural infection can prevent reinfection from variants. How long does this natural immunity last? We're not certain, but the data point to 1 or 2 years, which is not bad. But this natural immunity comes at too high a cost, right? Disease. Our goal in developing nasal vaccine is to replace infection with a safe nasal vaccine that gives you similar or hopefully better mucosal immunity. I do think nasal vaccines can work better than natural immunity. At our company, MESA, what we've done is we're developing a live viral intranasal COVID-19 vaccine that preclinically showed induction of mucosal immunity, systemic immunity, had high efficacy. We've enrolled approximately 100 subjects in a phase 1 clinical study. And thus far, we're seeing great safety with no what's called vaccine shedding and promising immune responses. So we're poised for acceleration. And our technology has been studied for another virus and 3 other phase 1 clinical trials. Look, we know from 200 years of vaccinology, going back to Jenner's smallpox vaccine, that vaccines that mimic natural infection tend to work best. They can be broad, they can be durable, they can block transmission. Okay, so this is our goal, but no one here today can tell you that mucosal COVID vaccines work. We're not there yet. We need clinical efficacy data to answer that question, and that's why we're here talking to you. So clinical efficacy plus immune correlate studies together can answer the questions, not just do these vaccines work, but how do they work? And that could not only potentially deliver this knockout blow to COVID, a transmission blocking vaccine, but also provide valuable information about vaccines to fight this virus and future pandemics.
Thanks very much. Now for something completely different, let's move from the nose to a new way to utilize the skin. And let's hear from the CSO of Micron, Mark Prausnitz, who will tell us about microneedles.
Well, I'd like to tell you about a different delivery technology, a technology that looks like this, a small patch that can be applied to the skin, a simple press, leave it in place a few minutes, peel it off, and discard it. We think that this can not only make vaccines more readily accessible, but also improve their immunogenicity. So I'm Mark Prausnitz. I'm representing Micron Biomedical today based on technology that was developed with support from NIH, NSF, CDC, Gates Foundation, and others at Georgia Tech, and has now been commercialized for the last few years via Micron Biomedical. So this patch targets vaccine delivery to the skin. The skin is another surface on our body. The body is equipped to mount immune responses especially well to pathogens that appear on the surfaces, because that's, of course, where they first appear. So what we and others have found is that if you vaccinate in the skin, you can get a more robust immune response as compared to vaccinating deeper in the body, such as in the muscle. Indeed, the only human disease that has been eradicated, smallpox, was done through vaccination in the skin. So in addition to that example, in the studies that have been done with other vaccines, what we have seen is longer lasting immune responses, broader immune responses, stronger immune responses, dose sparing enabled by skin vaccination. And for these reasons, we're optimistic that a skin vaccination can offer a number of immunological advantages. But what also drives this are the logistical advantages. You saw how simple it was to apply one of these patches. In fact, in the clinical trial we did on flu vaccination, we included a group where self-administration by the subjects was part of the study. They saw a few PowerPoint slides, they applied the patches, the results were just as good as when a nurse applied those patches. So we do envision a future where certainly administration by minimally trained personnel could be done of a special value in rural settings and in developing country settings, and potentially even self-administration when that's appropriate. These microneedles are as long as a piece of paper is thick. And so as a result, you barely feel them. They don't hurt. You don't see them. They don't look scary. And so as a result, acceptability of the vaccination should be higher. When the needles enter the skin, they contact the fluids inside the body where they dissolve. So you get the added benefit of a dry vaccine that is more thermostable. And we've shown for some vaccines that we can store them for 2 years at 40 degrees Celsius. So as hot as it gets in London a few days ago. And they're able to maintain that stability for years. But you don't have to redissolve it before you inject it. You take the dry formulation, you stick it on the skin, and it dissolves away in the skin, leaving no sharps waste. So that disposal is no longer, sharps waste disposal is no longer an issue. Manufacturing can also be done at a cost that's comparable to a pre-filled syringe. So we're optimistic about the future of vaccination that can be more accessible to people as well as be more immunogenic. Thanks.
Thanks very much. Now let's hear from Dr. Michelle Smith from the Arkansas Department of Health. Is this all going to make sense to people who are looking for a way to prevent illness?
Well, I hope so. And that's one of the reasons why I'm here. I'm also very excited to be on this panel. Thank you to the organizers. Because a public health professional, our job is to learn this information, digest it, and then put it out to the masses so that they can understand all of this fancy jargon and patches and everything. So as a reminder to public health professionals, I just want to encourage everyone to remember to always put public trust, trying to build public trust at the forefront of your work, because no matter how groundbreaking, how effective, how cool these patches are, if they aren't utilized, if people don't have access to them, then they won't do what they're designed to do, and that is to save lives and protect the economy. So how can we work from moving your advances from the lab and bringing them into American homes? First, I'm going to talk briefly about what Arkansas has been doing in terms of vaccine delivery. And then I want to highlight 3 Arkansans and highlight how these advances could help them. So first, to offer some context about Arkansas, our population is mostly rural. We have about 3 million people. We have high rate of comorbidities such as obesity, diabetes, cardiovascular disease. And before COVID, our office worked with non-traditional partners such as barbers, beauticians, fraternities, sororities, HBCUs, and faith-based network. So when COVID did come along, we were already well established in these networks. And so they could trust us already. And so we were able to quickly mobilize them to help us. They were like our boots on the ground. We would give them factual information about vaccines, about COVID, wearing masks. And we had town halls, webinars, fact sheets. And so it was great to already have that trust built into our network. So then they could take that information and then spread it out to individuals in their network. One example of how we used a health equity lens to vaccinate or ensure that vaccinations were accessible in minority populations is that we listened to a sorority when they called us and said that they had been calling the pharmacies and weren't able to get on a waiting list, much less make an appointment. So we listened to that. Based on that concern, we reached out to the Arkansas Pharmacists Association and we asked them to open more slots for minorities in their communities. And once that worked, we said we need to expand this. And so we developed health equity strike teams where we would go to communities, minority communities, and bring the vaccinations to them. We had teams in low vaccination counties, and we used lay people, we used pastors as volunteers. Because again, that trust, they knew someone, they were familiar with them, and that encouraged them to come get tested. Partnering with the Arkansas Pharmacists Association also gave us access to pharmacies that had these sub-zero refrigerators. A lot of the small pharmacies did not. And, you know, that's one of the advances that I'm excited about when you don't have to have that sub-zero temperature. This will allow us to go into more rural communities and vaccinate without having to have a network, which was good. We did have a network where the pharmacies would share with the others that didn't have that. But if they don't have to have that barrier, then we can vaccinate more individuals. So as a result of our health equity strike teams is what we call it, we had 127 vaccination clinics. And just from our team alone, we vaccinated over 19,000 minorities. Now, if I can, I want to just put a face to some of these numbers. I want to talk about some individuals and how they could have benefited from these things. First, I want to talk about Rosie Lucas. She was a community member who she actually helped us when we first started doing our testing. She called people, made sure they came out to get tested. Well, Rosie, who happens to be my aunt. Oh, okay. She was admitted to the hospital because she did not want to get vaccinated. She was, you know, a little leery of the needle. So some of these nasal injections, that this would definitely help her. And this impacted me because, of course, that was my family member. But I'm getting an eye over here, so maybe I can go back and talk about some of the other Arkansans that can benefit from this. But I want to end with a note to everyone up here to remember not only the adults, but the younger generation. I've come in contact with some kids that will blow your mind. And because they're watching the news, these types of things excite them. But we don't have a lot of diversity. And so I think that's another way to build trust. When we have 20 years from now, another panel like this, and we have African Americans, Latinos up here, then they can go into their network and talk about these advances. And then we'll have, greater access to these technologies.
Great. Thank you very much. Sorry for giving you the eye. So let's get into some questions here that I might throw at my panelists without having warned them, but these aren't too bad. One thing I think when it comes to the use of nasal immunization, the one that people are familiar with in terms of a human application is for influenza, and it's flu mist. And there's a general sense that it was okay, but maybe it wasn't quite as good as people had hoped. What did we learn from that? And is there some lesson there in terms of how you would apply this for COVID? Maybe I'll ask Dr. He to start that response.
And that's a really good question, because in animal, in veterinarian field, we've been using kind of cough vaccine for over 40 years now. It's been used really well, and there's great acceptance of the kind of cough vaccine, and they continue to be a vaccine that's been used. And for influenza vaccine, and I'm a big fan of flu mist, so some of you may have gotten it, and it's really a wonderful vaccine. And I think the lessons for me from scientific point of view to learn was that it went through a lot of difficulties in developing it. It's not because it's not a good vaccine, it's a wonderful vaccine, and you just get a spray in the nose. You don't have to get an injection, right? How easy is that? But the issue when you do something new, when you do something novel, right? It's exciting, but it's hard. Innovation is hard. I want to point this out. We have been doing this, as pointed out, for 20 some years now for intranasal vaccine, right? So the lessons we learned, and as in the early panel, we talked about what is immunocorrelate, right? In the case of influenza virus for flu mist and MedImmune, which later acquired by AstraZeneca. I mean, I know those people who worked on the project. They were fantastic, scientists. And yet, when they do this, they have been to deal with what is the immunocorrelate you can use, because it's a new vaccine. So traditional flu vaccine, we have a immunocorrelate. It's called HAI titer. And then you do that, you can basically very quickly get the approval. So when you do something new and innovative, and then what would be the immunocorrelate? Because this time you're using a live virus. And can you look at the IgA, or can you look at the the cellular immune response? All those kind of things which are critical for the efficacious effectiveness of flu mist, but there is no standard from FDA. So this is one of those things I hope when we move forward with the COVID vaccines for intranasal. I'm sure Marty was thinking about the same thing, right? And early on,
yes.
Yeah, what would be the criteria, right? What would be the immunocorrelate we're going to use to move this forward to get the approval, the BLA?
So you've raised it. Let me ask Marty, what would you say from your perspective would be most helpful here?
Sure. Let me back up and say, when flu mist was at its peak, it wasn't just an approved vaccine. It had a preferential recommendation in children over the flu shot. And the reason for that preferential recommendation is that it had better efficacy. And if you look at the data, what can we learn from flu mist? Well, it was more durable. And in the so-called school mist studies, it showed that it could reduce transmission. So these are the principles of mucosal immunization that we're aiming for. Now, what happened with flu mist? Obviously, it fell off. And I think without getting into the weeds, these are influenza specific and manufacturing issues. But at its peak, when it was made correctly, it was an outstanding vaccine, especially in children who are more seronegative to influenza.
Thanks. Helpful to have that clarified.
But the correlative protection question is a good one. So, this additional efficacy in children that we saw with flu mist did not correlate with the HAI titer, right? So there's something else. And that's when you're using a live viral vaccine, especially if it's mucosal, there's a double-edged sword. You're stimulating a broad immune response, innate, adaptive, cellular, you're not hanging everything on a serum antibody response. So it's more difficult to measure these mucosal immune responses. And I think Dr. Fauci brought that up in his initial presentation that, we need to better understand mucosal immunity. Well, we also just need more standard assays to study it, among us.
Yes.
So I think we could make some progress there.
And that was on his list that I quickly copied down of 7 barriers for making progress in mucosal application of vaccines. Let me ask Dr. Iwasaki, one of the things that I think you've written about is how it seems to work particularly well if you have a standard injectable vaccine and then you boost with a nasal vaccine. Can you say something about that? And for the case of COVID-19, where we hope a lot of people will have gotten that initial vaccine, is there something special there that we should pay attention to?
Yes, thank you very much for that question. Just to kind of round out the flu mist question, one of the issues about the flu mist and other sort of live attenuated pathogens is that once you develop enough antibody against the actual vaccine, the immunogenicity declines over time. So that's one issue that we need to keep in mind, that if we're going to be boosting over and over through the nasal route, we should be really paying attention to what kind of platform we're going to be using over the long run. And just to come back to Dr. Francis Collins' question, indeed, our approach is actually leveraging the pre-existing immunity that's generated by the intramuscular vaccines. And how it works is that you generate these memory T cell response that's circulating, and we leverage those T cell memory to serve as natural adjuvant. So most vaccines require both antigen and adjuvant. Antigen is the target that B cells and T cells see, and the adjuvant is the molecule that stimulates the innate immune response. Without the adjuvant, it's very difficult to engage adaptive immune response. However, what we found is that these memory T cells can serve the function of adjuvant altogether and not requiring any adjuvant to be delivered as a nasal spray when we do the prime and spike approach. And the other advantage, I guess, would be that because we can use the mRNA that's conjugated, complexed into a nanoparticle, that platform will not suffer from pre-existing immunity. There's no antibody against the nanoparticle or the mRNA itself to block that from becoming effective. So there are a few advantages. Every platform that we have here have pros and cons, and I'm just raising some of them.
That's really helpful. Let's talk again about microneedles. So Dr. Prausnitz, it sounds like there's a real promise here. I'm not sure I got a clear sense, though, about how this compares with intramuscular injection or nasal vaccination, if what you're trying to do, which is really important right now for COVID-19, is to prevent transmission. Do we have any way of guessing how that works for microneedles applied to the skin? What does that do for your mucosal immunity?
There have been studies comparing microneedle patch vaccination with the intramuscular vaccination, and those have been favorable. By some immunological measures, the responses are similar, and by other measures, you get a superior response when you vaccinate in the skin with the microneedle patch compared to intramuscular. I'm not aware of any comparisons between the microneedle patch and, say, nasal or wet mucosal vaccination. Indeed, the skin and the nasal mucosa have a similarity in that they're both surfaces and can leverage the immune system that is heightened on those surfaces. But the skin is not the nose, and if nasal transmission or pulmonary transmission is the mechanism, then the skin is not the site of entry for that virus. So it's unclear what role the microneedle patch might play in IgA or mucosal responses, but in terms of the systemic IgG responses, they are strong and favorable compared to intramuscular.
And let me ask about manufacturing. Seeing the microscopic pictures of these microneedle patches are pretty amazing. Is this something that is challenging to set up as far as a fabrication facility, or is this like no big deal?
Somewhere in between. So the way that we make them is we have a mold, we pour a solution on, let it dry, we pour another solution on, let it dry, and put it in a package. So the manufacturing process in principle is a simple one. It's one that pharma knows how to pour films and let them dry. There is the devil in the details. The manufacturing infrastructure does not exist today for mass production. It's under development. It should be available in the next few years, but the The expectation is that manufacturing can be done at low cost, at cost similar or maybe even less than a pre-filled syringe, but that manufacturing infrastructure does not in fact exist yet today.
One of those challenges on Tony's list, I think. Dr. Smith, I cut you off, but I think I want to hear another story or two about how this set of new technologies might be well received or not in populations that maybe have not been particularly well served by our healthcare system and maybe even are a little less trusting that the next thing that comes along is actually good for them. So say a bit more about that.
I really think that preventing transmission holds the most promise. Unfortunately, breakthrough transmissions are causing people to be complacent and just kind of lose hope in everything and just say, you know, what's the point? I'm still going to get infected. There was one individual, his name was Dale Smith. He first was hesitant to get vaccinated, but his wife got vaccinated and then he did. And he became a very strong advocate on social media. And he used that platform to kind of convince his friends, and he was a musician, other musicians, to get vaccinated, use masks. Unfortunately, Dale became ill, was hospitalized, and because of his one of his comorbidities, he died 5 days after being hospitalized. And so that gave a lot of ammunition to the naysayers that were already saying, you see, I told you I did, what's the point? So I really think that will help people once they see that this particular vaccine can stop transmission. I think that holds the most promise. But I also want to stress the importance of talking to individuals about what to do after they are infected. A lot of people in the minority community don't know about the treatments. We spend a lot of time talking about the science. We also need to spend time talking about therapeutics. Every American had the ability to receive the at-home test kit. We all need the ability to receive therapeutics the same way. And so, you know, we don't need to only focus on the science. We need to talk about, be honest about what these advances do and what they do not do. And then let people know that if you are infected, this is what you need to do to save your life.
Absolutely. It's a very good point. Dr. Iwasaki, I've read something about at least one company that's developing a pill for a vaccine. And obviously we know that has been successfully done for viral illnesses that we mostly associate with gastrointestinal problems like polio initially, or some of the diarrheal disorders. But at least there is some reason to believe that it might be possible to immunize orally and inspire, because that's also a mucosa, a response that could provide protection against infection and maybe also transmission. So how would you size that up? I'm asking you now as the person with the big, broad view of mucosal immunity as an alternative. And is there enough working going on in that space?
Yeah, that's a really great point. I think mucosal immunity elicited by an oral pill like the Vaxart vaccine is a very promising approach. In fact, we emit a lot of virus from the oral cavity. So if you really want to prevent transmission, you've got to have the defense system in the oral cavity as well as the nasal cavity. And I think it holds a lot of promise. I know that the phase 1 trial is showing promising data for durable IgE responses. I think we need multiple different approaches to be tried because, you know, we're not going to bet on just one thing to work for transmission blocking vaccines. The more we can test, the better. But I think we need the resource and the help to be able to get there.
And that brings us, yes, to what resources would be most helpful at this point. We talked about how nice it would be to have a clinical correlate that would be accepted by regulators as indication of an immune response that might be protective. And obviously, everybody has mentioned that more investment is needed here. In terms of other aspects, maybe just a quick round here. If you had to name one thing that is currently most needed to move your particular approach forward, Dr. He, what would you say? What do you need most?
Well, I guess what we really like to see to happen is that to have a more expedited clinical trial schedule and et cetera. And that's just beyond just, resources, money. We like to move faster. And which costs resources, takes resources.
It's all going to come down to money, I bet. But in your case, get the clinical trials up and going. Yes, Dr. Iwasaki.
Yes, for us, it's a practical issue, but we really need to be able to use the mRNA vaccine leftover in the vials for research purposes. Currently, we're not allowed to use all these discarded vials of vaccines for research purposes, and that's impeding our progress on multiple different laboratories. So if the CDC will allow us to use discarded vaccine for research purposes, that would be amazing. Thank you.
I would not have thought of that. Thank you. Yes, Dr. Moore.
Yeah, I think developing transmission blocking vaccines, of course, will come with great challenges and some cost, but I think the probability of success, warrants this investment based on what we know about vaccines, and the potential benefits are just enormous,
So
direct and indirect. So I agree with both Dr. He and Dr. Iwasaki. We need both R&D and clinical resources and coordination. We should work together to achieve this goal.
glad to hear that part. Dr. Prausnitz.
I'll agree that more clinical experience, more experience in humans is valuable. What I'll add on to that is topic of a moment ago, manufacturing infrastructure. We approached leadership both in the government and in private foundations at the beginning of the pandemic about what about a microneedle patch for COVID vaccination? And the response we got was, that sounds very powerful, but we need to move fast for this first generation of vaccines. And you don't have the manufacturing infrastructure today, and we need vaccines this year. So by having some of that infrastructure in place, we can be ready.
And maybe I, should ask our expert from the Department of Arkansas Public Health, what should we be doing in terms of the obstacle of public acceptance? Should we be sort of preparing for the possibility that we will not always be having needles going into arms?
Exactly. I mean, preparedness is basic 101, what we need to do. We need to have more discussions such as this. We need to take this on the road so people can become familiar with it before it actually hits the shelves. Also, social media is a powerful tool. TikTok, everyone loves that. You can create good content, but we just need to utilize faith-based networks, other non-traditional networks, to bring them to the table and talk to them to see what their misgivings or just barrier, what their perceptions are to some of these, and then go from there. But I think including people, everyone at the table early on is what's needed.
So a question from the audience. It's been noticed that the two companies that are pursuing nasal vaccines are working with intact virus, modified, killed, or weakened in some way, but not so much with things like mRNA or purified spike protein. How come? You can start, Dr. Herb.
Okay. So because it works, in my opinions. Like I said, we are really repurposing a kennel cough vaccine. A vaccine has been used in dogs for many, many years, 40 some years now. For the dog lovers, right? If it's good enough for the dogs, maybe it's good enough for human. So we have the joking aside, really for this vaccine system has been used for so long, and it has a great safety profile. When we develop a vaccine, really the number one issue is safety, right? Safe, efficacious, and cost effective. So this vaccine is safe because it's been used for so many years. Your dogs, when you come home, you will, come into close contact. Actually, vaccinated dogs can share a vaccine for a few days. And then, so you have seen this virus before in your life. So they're very safe. And of course, using a live virus has a lot of advantage over a dead one. In cell immunity and cellular immune response, mucosal, when you do the intranasal, and as well as a very good antibody response, right? So you have a very balanced immune response compared with just using one arm of our wonderful immune response. So, and again, because if you quickly Google kennel cough vaccine for a few dollars, you can buy it online. So think about if we develop a nasal vaccine for a few dollars, Amazon can deliver to your home. You can just spray it up your nose, right? And it's thermal stable. And then people, in the case of Dr. Smith mentioned, you have, instead of having people to sign up, you can deputize them. They can be the people to immunize using this device we have, right? Because this is already done with the docs. So we can, using the manufacturing capacity, the capability and all the things we have, that makes this an ideal vector system for developing nasal COVID vaccine. But again, this is just one approach. We hope, to succeed to address all the concerns that's raised.
Yeah,
but that's just one.
I think having had an opportunity many times to try to explain to people why the current COVID vaccines can't possibly give you the disease because it's a purified mRNA or a spike protein. Here you are talking about the complete virus, so you'll have to explain why it can't possibly cause trouble, maybe even in an immunocompromised person. I don't know. Dr. Moore, what would you say about using the intact virus versus other alternatives?
Yes, so I agree with Dr. He. Actually, he used the same list I was going to use, which is safety first, efficacy, and cost effectiveness, right? So we've already talked about oral polio, we've talked about smallpox, you mentioned rotavirus, we've talked about flu mist. All of these are live viral vaccines. And so, the challenge with the mucosa, especially in your nose, there's a lot of physiology there, right? There's mucus, there's ciliated cells. Typically, when you deliver particles, your nose is a filter. It's there to filter out those things. So live viruses tend historically to penetrate that kind of environment and deliver an immune response. So that's just, and I'm not saying it can't be done, but I think it requires innovation to deliver subunit or recombinant proteins to the mucosa. And those innovations are coming, but the most straightforward way to do it now is with a live virus. So then there's safety, right? So with our particular approach, we've taken great care over 10 years of research to highly attenuate our vector system so that it doesn't replicate to a high enough level to even shed in recipients. And I think it's important to start with that safety in mind first, because ultimately our goal is a global transmission blocking vaccine. And then cost effectiveness, I appreciate Dr. Reddy's comment in the first panel about, how dose can affect the COGS, the cost of goods, right?
Yes.
So compared to other types of vaccines, typically live viruses, you're using much less, essentially, biological dose, because the whole live virus gives you more potency. So that means you can manufacture at a much lower cost and begin to contemplate a needle-free global distributable vaccine.
So I've got to give Dr. Iwasaki a chance also to weigh in here, because I think in her opening remarks, she talked about using other strategies for mucosal immunity generation, including things that aren't complete viruses. So please.
Yes, thank you so much. So I absolutely agree with the two, Dr. He and Dr. Moore, on what was said about the barrier. Normally, if you want to generate a de novo nasal immune response, you do need a live infectious, attenuated particles in order to penetrate those layers and make enough antigen and enough adjuvant to be able to prime an immune response. What we found is that you can do that same thing without invoking a live attenuated pathogen, but just delivering spike protein in solution without adjuvant, or mRNA in a paste particle without adjuvant, because we're coming in with a pre-existing immune response that serves as an adjuvant itself. So this kind of strategy, we have to obviously show the effectiveness of this strategy in humans, but in animal models, it looks really promising. Gives you durable IgA response and very good protection from infection. So I think we are still in the development stage, and we need to be able to try all these different types of vaccines.
Excellent. So many flowers need to bloom. I see we're getting the high sign about the time. Let me ask each of you to give one sentence about what you would like this audience and those who are listening remotely to take away from our conversation. And again, maybe I'll start this time with Dr. Smith.
Well, I think my message is to have a wide net for your network, include everyone, especially people that you're trying to bring to the table or that you want to use your technologies. Make sure that they are there at the forefront. And also just think about diversity. And and utilize non-traditional healthcare partners like fraternities, sorority, HBCUs are an excellent resource.
Great. Thank you. Dr. Prosnitz.
I would like to say that there is a future of vaccines that goes beyond the intramuscular injection, a future that enables heightened immune responses based on targeting different tissues of the body, particularly superficial tissues, as well as delivery systems that are simple to use, that don't hurt, that are thermostable, and can facilitate increased access.
Thank you. Dr. Moore.
I think a transmission blocking vaccine is the way to get in front of variants and put COVID behind us.
Well said. Dr. Iwasaki.
I think, agree with you, Dr. Moore. Transmission blocking vaccine is really the way to stop the spread of the virus so that no more mutants can arise. And also, it's really the only way to kind of prevent diseases like long COVID, which I think happens because the virus spreads enough throughout the body that it can establish reservoir in different tissues. So I think not just transmission and infection blocking, but potentially long COVID blocking vaccines, that's really needed in the future.
That's a good point. And I know you're doing a lot of work on long COVID. Yes, Dr. Hu, you get the final word.
So I like to close with my optimism, as always, that I think we're going to have a vaccine, hopefully it's a vaccine, intranasal or the mucosal vaccine blocks transmission. And I know we're thinking about the human right now, all the transmission, but I also like to remind us, the COVID virus does get into animal, domestic, as well as wildlife. So we need a vaccine, not only for the practice of reducing infection in human, we really need a transmission blocking vaccine to block the transmission to animals, because if they become a reservoir, in the long run, we're going to have a problem, endless problem. So let's go get them. Let's go get the transmission block vaccine.
All right. With that image of vaccinating bats in China, let us thank our panel most sincerely for a wonderful presentation. And we're on to the next panel.
Hey, thanks, everyone. Hopefully you can all hear me okay. I feel like I need a guitar here to serenade you. Well, welcome, everyone. Hopefully it's okay with the organizers that I go ahead and get started. I know we're your last panel today. I have had the pleasure of tuning in online, so I'm taking advantage of the hybrid approach here, but it's really been pretty insightful to hear from all the various speakers beforehand. And I'm a bit biased, but I have to say I'm excited for this panel in particular. I think you you will be too once we get into it. I should let you know who I am. My name's Lois Pace. I am the Assistant Secretary for Global Affairs at our Department of Health and Human Services, and have been serving in that role for about 1.5 years. In this position, I get to see firsthand the value and the benefit of international R&D activities and investments worldwide. And it's really incredible to see the benefits that they do have, not just for vaccines, but also diagnostics and therapeutics. And so I just want to acknowledge the spectrum of innovations that are available, even though we're focused on just one today. So kudos to my colleagues who have spoken before me from FDA, NIH, BARDA, and elsewhere, as well as other parts of the US government who really do a lot of hard work working with partners worldwide to make sure these innovations are available. But importantly, is that availability and that access. I mean, innovations are incomplete without reaching the people that we're all hoping to serve. And so that's really the focus of this conversation right now. Without further ado, I will introduce our panelists. And thankfully, I think they're sitting in the order that I have on my paper, which is really helpful. But first, we have Dr. Richard Hatchett, who is CEO of CEPI, the Coalition for Epidemic Preparedness and Innovations. Hi, Richard, good to see you. And then we have Secretary Cody Kinsley, who is the Secretary of the Department of Health and Human Services in North Carolina. So it's good to see you, Secretary Kinsley. Thanks for joining us today. Seated next to Cody, we have Ambassador John Kangasong, who probably doesn't need much of an introduction, but he's our incoming coordinator for U.S. Global AIDS, our PEPFAR program at the U.S. Department of State. So it's very good to see him here in his current capacity. And then finally, we have Mrs. Denise Smith, who is the founding executive director of the National Association of Community Health Workers. And so please join me in welcoming them today. Very happy to have you all because you all bring a number of diverse perspectives when it comes to this important topic of equity and innovation and how innovations themselves can even serve to drive equity, whether that's in the government sector or outside of government, whether that's domestically or internationally, or whether it's top down or bottom up. And so hopefully we'll touch on each of those areas. But first, an opening question for each of you to respond. I really want to hear what you think has worked, what you think has worked well when it's come to closing this gap or bridging this gap between innovations and disparities or inequities. And then we'll use that to start our conversation. Maybe I will start with you, Richard.
Great. I'm just very quickly, CEPI is an organization that supports research and development. So I bring that bias to this conversation. We're also an international organization. So I would say that what has worked well throughout the pandemic is science and the scientific community, the sharing of information, the speed with which scientists and institutions pivoted to develop vaccines, delivered vaccines in record time. So the world did about as well as it was positioned to do. And I think that's an important caveat in delivering vaccines. What has not worked well is actually the topic of this discussion, which is getting vaccines to the people who need them. I would just say two recent analyses have estimated that vaccines probably save somewhere on the order of 20 million lives in the first year of their availability. But 20 million lives have also been estimated to have been lost during the COVID pandemic. And the majority of those lives were lost after vaccines became available. So we must do better on delivering to the people who need them as rapidly as possible.
Thanks, Dr. Hatchett. Appreciate you kicking us off. Secretary Kinsley.
So I think in North Carolina, what we found that really worked was being able to see where we needed to go. I think the greatest challenge we all continue to face is that we have a lot of kind of categorical descriptive information about the equity issues that we're trying to solve, but in real time, we don't have the data, the visibility into seeing it. North Carolina committed to building a vaccine registry where race and ethnicity and other demographic data was non-negotiable. We had to have that information, and then we could pair the visibility of that information to leverage, frankly, our providers and making sure that we could hold them accountable to vaccinate on par with the folks they were serving in their community.
And at the same time, pair that with community-based organizations that could leverage the trust that they long had to then make that. And in North Carolina, we saw it work. We closed the gap as far as vaccinating on par in our Latinx community, our Hispanic community, and we nearly closed the gap in our Black African-American community and did close it in several age groups. We have more to go. I mean, I think the challenge we're facing, and I'm excited to talk about today, is that our health system has a long history of inequity. And when you say you have a big problem, you need to move really fast, it's not going to get better. And so we have to make it a priority to bend it just as hard as we want to move just as fast.
Thanks, Cody. Dr. John.
Thanks, Lloyd. Let me start by saying my views here will be informed by my previous assignment, which was based at the African Union as the director of the Africa's Centers for Disease Control and Prevention. So what I'm going to say maybe sounds like an odd view, but I thought that what has worked from that perspective where I sat was the ability to use inequity to stimulate a dialogue in a resource-limited setting that says we have to manufacture vaccines, regionalizing the capabilities and capacity to manufacture vaccines as a health security commodity. I think I've always, when I left my previous job, one of the things that occurred to me was that the momentum is there to regionalize vaccine manufacturing capacity. We have to build on that, but do that in a way that you create an ecosystem of how to do it right, and not if it should be done. I think that is one thing that worked. What didn't work well, as Richard said, was translating the remarkable achievement and progress that science brought to all of us to making that accessible. And that is a complicated equation that has to do with manufacturing itself, has to do with human behavior, and has to do with the timeliness of where that product gets to the intended populations.
Thanks, Ambassador Nkangasong. Please.
Good afternoon, everyone. Denise Octavia Smith. I will also provide some context in that I have been a community health worker for about 24 years. I'm a certified patient navigator. I'm a survivor of a rare chronic disease. I am someone who has experienced homelessness. I am a person who has received public assistance. It's usually common for community health workers, promotoras, aunties, our outreach workers, our peers, to ground others in our lived experience. And so I'll be sharing some of that as I provide this context. Equity is not a top-down discussion. So we have to ground ourselves when we talk about equity today in a couple of concepts that I think are well understood in this room, that the systems, the policies, many of the funding models, the services were not designed for people who look like me, for those who are poor, for those with limited English proficiency, for those re-entering society or those of different genders, for those who are immigrants, refugees, or even for the indigenous nations, hundreds of which here in this country are not recognized by the federal government, and many hundreds who are, but who struggle with nearly every indicator of health and well-being that we have. It is important to ground ourselves in the understanding that it is not just about defining equity, that equity is a moral imperative, and that when we talk about equity, it's a call to action. So in the context of vaccines, I just want to say a couple of things. Number one, I want you to think about community health workers as being vaccinated. it occurred to me that many of us have traversed and overcome many of the barriers to the social determinants of health. We are survivors of rare chronic diseases. We are persons living with HIV. We are persons who have overcome homelessness. We are persons in recovery. And in that respect, we are experts in traversing those sectorial barriers, those literacy barriers, those technology or navigation barriers. We are in fact vaccinated, but we have a commitment to go and help others, right? So what worked well? One thing that I really want to point out that is important is that in nearly every single major initiative or strategy that the Biden-Harris administration released since January of 2021, it has prioritized, it has included community health workers, right? So in the national COVID response and the preparedness plan, when we talk about mental health and in the American Rescue Plan, what we didn't do well is recognize that we needed to build not only a clinical response to the pandemic, we needed a public health response to the pandemic, but we needed the infrastructure, capacity, resource, and funding for a community-based response to the pandemic. And so with all that I've heard today and all that I'm excited about in terms of the technologies and the innovations, I don't want us to build on a faulty foundation. Community will bring these vaccines to the last mile. And further, we will make sure that the barriers to the social determinants of health do not impede vaccines getting into people's arms or through nasal or all of the other exciting innovations.
Thanks very much to each of you for getting us started. And I'm even more excited for what's to come. So I do want to start with you, Mrs. Smith, because I know something that you just mentioned about what you're hearing and how you're interpreting that. I know that you can't speak for all community health workers, right? To your point, everyone has their own experience, but I'm hoping that you can share perhaps what you are hearing from them with regards to these new innovations, if anything, right? What's breaking through and what are the things that are sort of exciting them about these prospects or keeping them up at night?
Well, you know, part of what's keeping us up at night is that the current vaccines have not gotten to everyone. We are very concerned about our children in particular, our babies, but generally speaking, we know that childhood vaccinations generally have really waned over the past couple of years. And we are seeing adult vaccinations, which have never been fantastic, also having continued barriers and just being lumped into this general sense of misinformation, right? And so a lot of our work is about dispelling myths and making sure that people have factual information to make the best decisions. But I'll tell you what is really keeping CHW or promotora leaders, those who lead organizations in the community, is that a lot of our funding for our work is actually shutting down right now. So there was a great story in March by Kaiser Health News about 400 CHWs in Chicago who had been trained, right, hired, recruited, embedded into a vaccine response. And by June, their jobs were gone. Right? That's an example of an infrastructure, dollars that are wasted, time that is wasted, expertise, relationships, partnerships, and it erodes trust in community. So what we want to do is we want to be a part of the solution and a part of this continuum from the science to the streets, if I will. And so we need that funding infrastructure to sustain these discussions.
Thanks very much. Cody, I want to come to you if that's okay. I'm sticking with the theme of sort of local and state level options. Denise mentioned very briefly the role of community health workers in messaging or messages. And I know that you have done a lot in the state of North Carolina in that regard. So can you give us some sort of preview as to how you would even talk about this work and maybe even blending what you just offered, right? Recognizing there's still some road to travel with current vaccines and vaccinations, and yet with others coming down the pipe.
Yeah. So I'll offer kind of maybe two examples of things we did in North Carolina and we're still sort of testing. So one is North Carolina created a counseling code as part of our Medicaid program. I believe one of the first Medicaid programs in the country to do that, and made it so that, you know, we found that as we got through the vaccine eager into the vaccine hesitant, it took many conversations for that. I mean, sometimes it was could have been the insured Medicaid child, but it was the conversation with the parent. And so shaping the code such that we could fund that, I think to Denise's point about being able to pay for what we actually want to see. And what's interesting in North Carolina is we've started to do a cursory review of that is we've actually started to see uptake of vaccines in kids increasing, while we're seeing some of that, not just for COVID, but broadly, because we see our providers really making use of that. So I think that's a very tactical example of some exciting work there. I will perhaps just go even double down on Denise's very good point, which is that we have invested so much money in community-based organizations that are doing on the ground work to leverage trust, to grow, to establish routine and stable communication channels, to trust folks. And my real and tactical problem in this moment is that I want to use some of that for monkeypox, and I can't because those dollars aren't monkeypox dollars. And I think we have got to wrestle with what I call color of money problem in government. And if we're going to invest in the foundation of this on the ground stuff, we have got to sustain this over time.
Very interesting points from you both. Cody, can I ask a follow-up question of you, which is this idea of how we do leverage what's in place? I mean, do you foresee an opportunity with mass vaccinations with these innovative products, or is it your worry that you won't be able to get those kinds of campaigns off the ground in the future?
Well, I mean, I think that, when we have a whole lot of folks coming forward in that kind of vaccine eager space or test ready space, obviously these kind of mass distribution sites work really well. I mean, the kind of dollar per person operational effort is efficient and it works. I mean, of course, right now we're all pivoting towards an at-home strategy, right? I mean, we have to reduce the number of drive-up testing sites in North Carolina towards a place where we're leveraging at-home pickup sites. And we're obviously, thanks for the Postal Service and all of that. And so what I'm excited about some of the conversations about these more novel delivery methods is how can we reduce the overall burden on the healthcare system? Because the other side of this is that our healthcare providers are so tired. Everybody is spent, our workforce is incredibly drained. If we can try to minimize the impact in those spaces safely, I think that there's a lot of promise for some of these more novel delivery mechanisms. But we have to think about that kind of end-to-end effort that it will take to do that.
Thanks very much for taking that follow-up. Dr. John, I want to come back to you actually on this topic of how we innovate the delivery, right? And reduce the burden on healthcare systems, not demand so much of the systems or the citizens for that matter. I know that you in your current role are overseeing the US's global AIDS programs, and a lot of that work through our President's Emergency Plan for AIDS Relief, a lot of that work has been leveraged in the global COVID response. So what's your take on how we can worldwide ensure the delivery of these new vaccines, especially given your experience in PEPFAR and new innovations for HIV?
No, absolutely. I think Denise and Cody also touched on this. Delivery will always be human capital related. And that will require that we leverage on what is already available. When I hear discussions about pandemic preparedness and response that hover around creating some new mechanisms on to put assets together, I said that in my view is the wrong way to go. I mean, we should be looking at how platforms that we have invested over decades, like the PEPFAR platform, can be elevated and used more appropriately to deliver, which is essentially supply chain management issues, workforce issues, information system issues, which already exist. And PEPFAR did some of that, quite a lot of that to support many countries in Africa, where you saw vaccination rates for COVID increase significantly when those platforms were appropriately leveraged. I think that those are kind of things that we should be looking at, including community healthcare workers that played a very important role, including during the early phase of the COVID response, where we just didn't have vaccines. It was, we were fighting it with the very basic public health measures, which was test and trace. And you deployed at that time, I recall, using about 20,000 community healthcare workers. South Africa did that very successfully. And most of those community healthcare workers are those that had been trained on HIV. So I think the task ahead of us here is really not to create entirely new systems, because they will go to waste, but to leverage a system that are already being used. I think that is very, very important. So I'll just end by saying that elevating these platforms that we've all known that deliver in a very impactful manner is the way to go, not just for PEPFAR, but the global fund as well.
Well, thanks very much. And I see Denise nodding along, so it just shows the synergies here. Richard, I want to come to you because I've heard you talk about the importance of inserting equity at the beginning, right? And I know that the work of CEPI is really focused on that, really looking at the pipeline from the start. So can you explain how that's getting inserted into how you make your investments and decisions?
Yeah, no, thank you. Thank you, Lois. I was actually eager to make the observation that, here we are talking about equity at the end of an interesting day. And, but that's not how we should be thinking about equity. We should be starting with equity. Equity is not a discrete goal that can be attached on to other discrete goals. It has to be a mindset, and preparedness actually also has to be a mindset, and it has to inform all of the decisions that you make. Just to illustrate a little bit about what that meant in terms of CEPI's response, our institutional response. CEPI was created to support vaccine development. We were actually created to address epidemic diseases. Initially, we weren't considered to be a pandemic response organization, but when COVID emerged, we jumped in. And we jumped in by moving fast because we were concerned about the potential for global spread. But we also were concerned about equity. We anticipated, based on our experiences during the last pandemic, which in 2009, which fortunately it was a very mild virus, but the potential for global catastrophe was large because we did not achieve equity. And we did not distribute vaccines globally until quite late in the pandemic. So as we began to gear up to develop vaccines, we actually wrote into our R&D contracts requirements for equitable access. And we actually envisioned, with our colleagues at Gavi and WHO and ultimately UNICEF, envisioned a global allocation mechanism which became COVAX. And even before COVAX existed or had a name, we were writing contracts that were requiring our partners to make doses available for the global allocation facility. And I would just say that the root causes of the inequity that we experienced in 2021 in particular, after vaccines became available and before supplies became robust, were manifold. There were things relating to the technology. I mean, cold chain requirements for the vaccine were an issue. There were institutional causes. There were financing causes. There were structural causes. I would say John mentioned the way vaccine manufacturing is distributed globally. And to address equity as a whole, we have to bring an equity mindset to thinking about each layer of those problems, both at the sort of holistic international level, all the way down to the delivery and breaking through these barriers that specific communities or subsets of communities may encounter. And so that is how we at CEPI have incorporated this idea of sort of equity first, and then make decisions that lead towards that goal.
Thanks, Richard. I'm hoping I can ask you a follow-up question as well, which is whether or how you all, as part of discussions around COVAX or the broader ACT Accelerator, how you all are thinking about next generation innovations and how that applies to what's been established or what's to come.
Well, so COVAX is the international mechanism for procuring and taking vaccine donations and then delivering those globally, just for anyone who's not familiar with it. There were a tremendous number of innovations, that ad hoc innovations, essentially to solve problems in real time as we went along in terms of procurement, structuring advanced purchase agreements, setting up a global liability and indemnification system and a no-fault compensation system that facilitated rapidly, making vaccines available in different countries and different legal environments. We've developed an inventory of those innovations, and again, they are manifold across different domains that had to be addressed for COVAX to operate. And we've thought about which of those innovations are sort of intellectual innovations that you can, as the pandemic winds down or as we shift into a different mode, how you can bundle those up and have them kind of waiting to be called out again. I mean, one example would be the allocation framework that WHO developed. That's something that in a future pandemic, we could bring down as a place to start. Other innovations or other areas where innovation is required, where we ran into really significant challenges, are going to require ongoing work. Financing is one obvious example. Some of that work has already begun. The last area are innovations that actually now need to be embedded in day-to-day practice and in day-to-day delivery and day-to-day communication with communities. And so we've begun to think systematically about how we can capture the innovations, but also the lessons learned, and then think about how we can make those sustainable for the long term.
Thanks very much, Dr. Hatchett. Okay, back to you, John, because you are doing a lot and have done a lot in terms of rallying sort of this leadership at a regional level. And so I'm also wondering what you're thinking about these innovations and how, in particular, not just global platforms like COVAX, but regional platforms such as what's come together through the African Union or other parts of the world can also best take advantage of these new products and technologies.
Absolutely. Let me just reflect a little bit on COVAX, and then I'll segue to the regional question. At the height of the pandemic, I thought COVAX was one of the greatest innovations in recent times in global health that are equate to the innovation in Global Fund, the establishment of the Global Fund. It was one of the greatest, the greatest good intention to solve a global problem collectively. And it also represented, in my view, a great symbol of solidarity and cooperation. But how that intent and all these things translated into practicality were challenged. I think we should also learn from that. And challenged not because of COVID, but because of externalities that surrounded the whole. So we should build on that and try to fix it and look at it in a holistic way. And this is where I segue to the regional platforms and truly have a mindset that listens. A mindset that listens to the regional aspiration of what different parts of the world are trying to do. As we speak, the ASEAN countries are establishing a public health agency. What the PAHO, the Caribbeans, the African regions, the Gulf states are establishing a citizen. So that says something that we should be thinking of our global health security architecture at three levels. I mean, what do we do globally? What do we do regionally, and what do you do at national level? That is a collection of that network that will give us that health security instruments that we are looking at. When you look at regionally, at least from where I just spent 2.5 years until last April, the continent of Africa rallied around the political leadership and goodwill and said, look, despite COVAX, we're going to create an African vaccine acquisition platform and secure 400 million doses of vaccine. That is not opposed to the central COVAX mechanism, but rather complementary. That those are kind of things that you need to encourage more. We need to encourage more. And as we think of the next generation vaccines, begin to see how those platforms that were put in place regionally can enable us to leapfrog any challenges that were observed during with the issues with the first generation vaccines. Let me just say this, that in Dr. Fauci's opening statement, he talked about leveraging the HIV, that some of the advances that we saw in HIV, in the COVID vaccines were because of the networks that we have established from HIV. It's also true as we regionalize and begin to mitigate the risk of putting all our eggs in one basket. I think that is exactly what we should be looking at. How do you learn from the experience in a positive way, okay, and have the courage of saying there were some gaps that occurred. We should build on those gaps to strengthen our collective security apparatus, including support of the regional initiatives.
I'm curious from you, John, before you turn off your mic, how you're also thinking about opportunities for regional manufacturing. These are investments that even the US has made alongside other countries. I think to your point, there's an opportunity, obviously,